Hello, everyone. Welcome to our 50th Infogest Viva Webinar on food digestion. Today, Ruoxuan Deng will present part of her thesis entitled A Live Show in the Stomach, Monitoring in Vitro and In Vivo Human Gastric Digestion with MRI. Ruoxuan obtained her PhD at Wageningen University last year, and now she's working as a postdoc at Agronomic Institute INRA in France. She won the prize for the best presentation at the International Conference on Food Digestion in Cork last year. The webinar is recorded on YouTube Infogest channel. Ruoxuan, thank you for being here today. The floor is yours. I'm going to share your video.
Hello everyone, I am Ruoxuan Deng. I'm a postdoc researcher in Teagasc and Infogest in France. Today is my great honor to present you about my PhD thesis in which I did in Wageningen University, entitled A Live Show in the Stomach, Monitoring in Vitro and In Vivo Human Gastric Digestion with MRI. Perhaps like many others, I'm super passionate about delicious food. When I was young, I kept imagining what would happen to all types of tasty food when they enter into my tiny stomach. I couldn't get an answer when I thought of it. Although I know my stomach was very happy and I was very energetic after I consumed all this nice food. But luckily, afterwards, long time after, I started to do research. I worked on gastric digestion. So I guess many of you would agree with me that digestion is super important process in human body. Among the digestive tract, gastric digestion is a crucial part because it's an organ that would involve physical breakdown of the food particle because of muscular construction in the stomach, as well as it involves chemical degradation of food because of the presence of gastric acid and enzymes such as pepsin, lipids, etc. So food particles can only enter to next digestive stage when it's smaller enough. The crucial part of the gastric digestion is that it can reduce the particle size, break down the food structure, and it's also important, especially for products with protein, because gastric digestion is the starting point of protein digestion because of the pepsin and acid. To study gastric digestion, thanks to all the researchers in this field, there are already a lot of very useful in vitro gastric digestion models available. With these in vitro models, we could analyze how the food property changes during digestion and what's happening in the digestion fluid, as well as the interaction between them. So, using in vitro models, we can withdraw sample from it and do the analysis as we want. Apparently, there are many advantages of the in vitro digestion model. It is a very well-controlled system and is much simplified compared to the real condition. We could test more straightforward on the research question and get an answer in a fast way. There are all these advantages of the in vitro models. However, these models also have their limitation. Because when you think of the human body, actually when we are simplified the human body into the models, we limit the models to represent the biological variation and the complexity of the human body. Plus, it's very difficult to use in vitro models to represent the feedback mechanism happening in human body, such as hormone response, neural system, etc. Actually, there are already some approaches that are available to study in vivo on gastric digestion. For example, gastric aspirate, we could take sample from stomach and do the analysis, or taking blood sample to measure nutrients and hormones, or ask the participant to read their appetite. However, this approach is either too invasive or indirect. There is a question that whether there are some methods that could help us to link in vitro and in vivo digestion and help us to validate the in vitro models. Probably you know the answer already. Here we propose that MRI might be the right candidate that can help us to understand better what's happening in vivo and help us to link the in vitro and in vivo data on gastric digestion. And that was what I worked for during my PhD. For those who are not so familiar with MRI, it is an abbreviation of magnetic resonance imaging, and this is how it looked like. This was me when I was conducting my human trial. Now you may have the question, why MRI can be the candidate? Here I'm showing you some MRI scans of one participant before and after drinking a glass of milk. At the left is before drinking milk, so it's after overnight fasting. Here you can see the stomach is very tiny, and at the right side is after drinking a glass of milk, and the stomach region is clearly bigger. With this type of scan, we could analyze the volume change during digestion so that we could get information about gastric emptying. Besides, we could take a close look at what's happening in the stomach region, and we might discover if there are any intragastric processes happening, such as mixing, phase separation, coagulation, etc. Apart from using MRI as a camera, we also propose that there are some potential MRI parameters might serve as a very useful digestion markers to indicate the digestion of or hydrolysis of nutrients, so that we can make use of these MRI parameters to indicate digestion more quantitatively. There are many of them which are very interesting to discover. During my PhD thesis, I focused on the T1 and T2 relaxation. So I will also show you about it in the following presentation. What are T1 and T2? Today I won't go too much details into physics, but I would love to let you know how we can make use of them to indicate digestion. They are based on proto-relaxation. T1 describes the relaxation behavior of Z direction, which is called longitudinal relaxation time, while T2 is called transverse relaxation time, which describes the relaxation behavior in XY plane. Why they can be useful for indicating digestion? Here I draw the T2 relaxation curve of gastric digestion fluid and protein gels. Already from the relaxation curve, we can find that they are very different from each other. With this curve, we could calculate the T2 value of the protein gel and gastric fluid. Here, the difference is obviously big. With this, it gives the potential that we can use T1 or T2 to look at different components in digestion system. So what are the factors affecting T1 and T2 value? To make it easy to understand, you could think of that T2 and T1 are dependent on the proto environment. So if the proto is in a very free environment, like gastric fluid, then the relaxation time is long. However, if the proto is in a more rigid environment, such as protein gels, then you would get a shorter T2. So there are actually many factors affecting T1 and T2, such as concentration of macromolecules in the system, food structure, pH, or water distribution and water content. All these factors are existing in the digestion system. Also, they are changing during digestion, which makes T1 and T2 very attractive or very interesting to explore because they might be able to indicate these changes during digestion. However, it also gives a challenge that it might be difficult to interpret T1 and T2 because of all these factors would affect the values of them. Therefore, to explore whether we can make use of these MRI markers, we decided to start with a very simple digestion model. In the beginning of my PhD, we started with static digestion model, where everything is well controlled. We use whey protein gels as model food and add simulated gastric fluid in it. We did the digestion in an MRI and measure it from time to time. So this is how it looked like after 10 minutes. With time going on, something is happening. So here is a scan of the T2 map after 90 minutes. If you look at the supernatant phase, you would find that the T2 value gets shorter. This is because of protein digestion is happening. Because as I mentioned before, this is a well-controlled system, so we know that pH remain no change here. So what happened here is that because of protein digestion, more protein present in the supernatant phase. We measure the protein concentration in the supernatant. We also measure the T2 value in the supernatant. We find a very nice association between MRI markers, which is the T2 value, and the degree of protein digestion. This finding was so promising because it confirmed our hypothesis that the MRI markers indeed can be used to monitor protein digestion in a static gastric digestion model. However, now you might have a question that we all know in reality it is more complex than the static digestion model. The pH must be changing over time in the stomach, and there is gastric juice segregation all the time, and there is gastric emptying happening as well. So whether the markers can still be useful if the condition is more dynamic. With this question, we move to the next phase. So in the next step in my PhD, we develop a MRI compatible semi-dynamic gastric model, Mr. Gas. With this model, we want to examine how MRI markers can be useful if the situation is more dynamic. So with this model, we have the gastric segregation unit where we could add fresh gastric fluid into the stomach chamber. We also add the gastric empty unit to make it better representing the human complexity. The good thing of this model is that we could put this stomach chamber in MRI, and we could do the digestion in the MRI, and we could do the scanning during digestion, during the semi-dynamic digestion. So what did we find from this experiment? Here I'm showing you the readout from the Mr. Gas model. In the picture here, you can see is the T1 and T2 maps, the two MRI markers that we measured. With time going on, as you can tell from baseline, which is before digestion, till 90 minutes digestion, the T1 and T2 of supernatant change over time. So here we measure the T1 and T2 of supernatant over time. We also measure the pH and protein concentration in the supernatant. We build an equation models combining the T1 and T2 and the protein concentration and pH. What we find is that in this model, if we use the markers we measured, so the values we measured with T1 and T2, these two MRI markers, we could be able to estimate the protein concentration and pH real time. and the estimation are quite good and quite representing the measured value of protein concentration and pH during digestion. This readout showed us that even though it's in a more dynamic system, we can still make use of T1 and T2, these two MRI markers, to predict the changes in protein concentration and pH during digestion. These findings convince us how useful the markers are and give us confidence to dive in further to conduct a human trial to test how these markers would behave in vivo. So then we designed a randomized crossover human trial called We Digest study. In this study, we included 18 healthy participants, and we provide 3 treatments to all the participants. So they need to visit us 3 times to consume the 3 different treatments, which are 3 types of protein gels plus water. The gels are either differ in protein content or differ in hardness. So they are soft, low protein content, hard, low protein content, hard, high protein content. So we provide these 3 gels in 3 different sessions. In each session, the participant come to visit us after overnight fasting. And then we measure a baseline scan and we ask the participant to rate their appetite. Afterwards, we give the participant the protein gel to consume. So this is how it looked like. You probably already noticed that here is a webcam because we use this to record the eating behavior of the participant. After they consume the protein gels, which is within 10 minutes, then we put them back into MRI scanner. Every 10 minutes until 85 minutes, we did the MRI scan and we asked them to rate their appetite. At the end of the session, we provide the participant with an ad libitum meal, so we could measure in the end the meal intake, so that we can see the ad libitum intake if that's related with the appetite rating and the gastric volume during digestion. For the MRI scans, we take different types of scans. We took the anatomical scans, which I showed you early in the presentation, to measure the gastric empty. We also measure the T1 and T2 maps. Here I would like to show you some results. First, we took the scan, the anatomical scan, where you can see the stomach clearly, and we have these multiple slices covering the whole range of stomach so that we can measure the gastric volume. And this is a readout of the gastric volume over time. So in the beginning, the volume is very low. It's because there is only baseline basal gastric acid. Afterwards, after consumption of protein gels, clearly the gastric content volume increased because of the presence of protein gels. Gradually, because of gastric emptying, the stomach content volume decreased again. When you compare different treatments, we would find that gels with higher protein content and with a harder structure had the highest gastric content volume over time, which means that indeed the harder structure and higher protein content would delay the gastric emptying. Apart from observing the gastric emptying, we also measure the T1 and T2 markers. Here I would like to show you the example of T2 maps. T2 maps are in sagittal direction. Here is the back of the participant, here is belly. This part is kidney and this part is heart. The region within the white line is the stomach region. Again, the maps are color-coded, the lighter color representing a higher T2 value. So with this type of map, we could analyze the average T2 value within the stomach region and plot the average T2 value against the digestion time, which is shown in this figure. So the y-axis is the average T2 in the stomach region, x-axis is digestion time. As you can see at baseline time point, the T2 value is very long. If you still remember what I shared with you before, when the proto is in an environment which is very free, then you would have a higher T2 value. So it makes sense at baseline time point the T2 value is high because there were only basal gastric fluid. At 15 minutes, participant already consumed protein gel, and that's why the T2 value decreased a lot. With time going on during digestion, T2 increased again. This is probably because during digestion, gastric segregation taking place and gastric emptying took place as well, with the digestion progressing. So the local protein concentration in the stomach region decreased over the course of digestion, and that's why T2 increased. And this is the same trend for all three different treatments. Well, when you compare the treatment, you would find that soft low protein content, this kind of gel, always has a higher T2 value over the course of digestion in the stomach region. This is probably because with the soft low protein concentration gel digested faster compared to other two protein gels. And that's why it would have higher T2 value over time during digestion. So it's very interesting that this result showed, even though in a very dynamic and in a complex reality, the T2 markers, the T2 values, can still be related with the progression of digestion. So indeed, the MRI markers could provide us with additional information on digestion. When you compare the analysis of the in vivo scans with the in vitro experiment we did before, you could find that here we are analyzing the average T2 value in the stomach region. However, in the in vitro scans, we were able to measure the average of the stomach content, or the supernatant phase, or the gel phase. So we could get more clear information from the in vitro scan. This is because of indeed in vivo is more messy compared to the in vitro scan, and we have to compromise with resolution as well to get the sequence which could fit the limitation of breath hold, etc. So there are limitations when measuring in vivo MRI or when interpreting the data. There are rooms for future investigation on improving the sequence and also on how we could interpret the data in a better way to get more information from the in vivo scan. But still, the in vivo measurement we had in this study showed that we could get additional information based on the T2 markers, and the T2 values in vivo are also associated with the digestion process of protein gels. So the in vivo T1 and T2, the in vivo MRI markers, are very valuable to compare with the in vitro data and to link with the in vitro result. So I've shown you part of my PhD work in Wageningen University. With the presentation today, I hope I have shared this information to you. MRI techniques are promising method to study digestion because they can not only present the intragastric processes of food, but also provide quantitative information on nutrient hydrolysis. my research focused on protein digestion, but I believe that this method can be also applied on other nutrients like starch, lipid, etc. But it needs more investigation. Also, it would be nice to move further to more complex food with MRI technique. And probably in the future, we can apply this technique to measure digestion in different population as well. So there are many research questions might be answered with use of MRI technique. And with the in vivo MRI measurement, it can help us to link the in vitro result with in vivo data, and it might be useful to make use of the in vivo MRI result to validate in vitro and in silico digestion models. With this, I would like to thank all my PhD supervisors in Wageningen and people who helped me during my PhD. And of course, I would like to thank you all for your attention.
Hello, everyone.
Hi, Roxanne. I will invite you to turn on your camera and mic. Thank you really much for your presentation. Really exciting work in MRI. A lot of work, surely, around these images that we are seeing here. I will invite all the attendees. We have 50 participants at the moment, which is pretty good, that they can ask questions to Ruoxuan using the box, the Q&A at the bottom of the screen. For the moment, we have still a few questions, Ruoxuan, and I'll start with a question from Shaktivesh. Was the colon also MRI during this study? So, or is it possible to do a T2 map of colonic content?
Yes, thank you so much. Thank you for organizing this webinar, and I'm very happy to be here. And also thank you for the question. In current study, in the previous study, we didn't measure colon with MRI. So we didn't, the focus was only in the stomach. But I guess it would be possible to do T2 map of the colon. The only thing is that we need to consider about the size, because stomach has larger content when you do a cross section. However, colon it's much smaller. So in that sense, we need to be careful with the resolution and see if we can see the content that you want from the colon. But I guess it should be possible, but we need to do careful investigation on that.
Okay, so would you have an idea about the resolution of MRI, kind of a minimum diameter that we can obtain?
Yeah, that's again very good question, because when talking about MRI images, we always make some trade-off, because if you want to have high resolution, it can be possible, but it requires longer sequence or more energy input, which will be sometimes it will be limited when you are doing a human study, because you might need to ask the participant to hold his breath when you do the scan. So the scanning, there are more limitations on scanning sequence. In my previous study, the resolution of the T2 maps, it's around 2 millimeter square. So the voxel size is like that. So it's not very small. I think if you think of the size of your food particle. So therefore, I think in the future, we really need to think carefully what research question is and what you're looking at, considering the resolution of the imagings.
Yeah, so resolution is the issue. And we have another question about comment and question about resolution. What's the time resolution of the MRI, meaning frame rate?
Oh, yes, thank you for the question. So to clarify what we are talking about the resolution is that, actually we're talking about how clear the image is. So if, for example, if you have a photo of a person. If I have a photo of myself, if the resolution is very high, then you can clearly see everything on my face, every hair, or I don't know. But if you have a very blurry image, then you have a low resolution. So that's what we are talking about for resolution.
Okay, it's really clear answer. Thank you for that. And another question, thank you for the assistance here, from Tijal, who said, what materials did you make your in vitro stomach out of? And how did you validate your model?
Thanks for the question. Because I suppose you are talking about the Teagasc. So the model we built for the semi-dynamic MRI compatible digestion model. I suppose you're talking about that one. With that one, we use the glass as a compartment. So it's actually a water jacket beaker where we can have the water going from the outside to make the temperature at 37 when we do the digestion in the scanner. So it's we have to admit that because this is the first digestion model that we could put in MRI. So it's quite far away from the very fancy model. So this model was our first try. So the material is just made with the glass beaker. And then with that model, we took reference of previous dynamic model. For example, the gastric emptying rate or saturation rate, we took the values from literature. So that's how we built that model.
Okay, thank you for that. Keeping the talk about the model, regarding the relaxation parameters, so T1, T2, you analyzed at T10 and T90 minutes. Okay. Why did you use these times? Did you have some preliminary tests to determine them?
Thank you for the question. So I think this question was talking about the first study we did. So when I did this static in vitro digestion, I show you in the picture of T10, so 10 minutes and 90 minutes as the example. But actually we measured from before digestion to, I think, 2 hours also. So I just showed these two pictures because it can clearly show at the beginning of digestion and in the end of digestion, something has happened. So in a T2 map, we clearly see the color changed. So the content was changing. But indeed, we took a longer period of time to see what's happening. And we took this time because of recommendation from Infogest that the gastric digestion can be 2 hours. So that's why we took this time frame.
Thank you. We'll move on the next study with protein gel this time that you used. And people are asking what kind of protein was used to prepare the gel for the human trial, and which kind of protein concentration range did you use?
Thanks. I could have made it more clear in my presentation. I forgot to mention, but we use whey protein isolate as a material for making the gels. And then it's throughout my thesis, I'm a good friend with whey protein. So it's always whey protein isolate as the material to make the gels. And for the concentration of the gels, we chose 13% of whey protein and 17%. We did some trying on what the concentration we should choose for these gels. We want to make good contrast between concentration, but also making good contrast with the food structure, which means hardness. So we use these two concentration to reach a quite different or quite distinct hardness, so that we can have these two different groups of treatment.
Okay, and about the different gel strength, sorry. How much of the variability in the T2 from the in vivo study could be attributed to the different gel strength?
So I guess the question, thank you again for the question. I think it's a very nice question. I think if I understand correctly, this question is asking about whether the different hardness of the gel would bring us the different T2 in the in vivo study.
Exactly.
Yes.
Yeah.
So So we did measure the gel also before. So with pure gel, we measure the T2 value of them. I think the variation is between the T2 of the gel is quite short compared to the liquid. So it's around 100 millisecond to 200 millisecond. So it's within that variation, the T2 values.
Okay, really clear. Thank you. Talita, maybe you want to take over for the next question.
Yeah, we have another question here, very interesting. How possible is to understand and measure the viscosity of the contents with MIR? And how possible is to image complex meals like FDA breakfast meals? Is it possible?
Thank you. Indeed, this is again a very interesting question. Actually, I think a long time ago, it's around, I don't know, I don't remember which year it is. I think it's around 2000, like 20 years ago in Nottingham University, in a group of Luca Massiani. They did some work already on using T2 and T1, like MRI, to measure viscosity of food or of samples. So I guess you can try to search for that paper. Otherwise, you can send me an email. I will give that paper to you. And I think another question is about how we can image the complex food with MRI, right?
Yeah.
I think it would be possible if you are doing anatomical scans and then looking at what's happening in the intragastric processes. It should be straightforward because you can look at the content over time. However, we have to admit that it must be very challenging if you want to understand, or if you want to use the T1 or T2 map to know hydrolysis of nutrients, for example, in this complex content. So I think to that, we still need some work to investigate so that we can understand better things in a more quantitative way. I hope that answered this question.
Yeah, thank you so much. We also have another question here about the proteins part.
Oh, yeah. Yeah, sure.
They are asking regarding the graphic MIR marker versus the degree of protein digestion. How did you measure the protein digestion? By OPA? And did you analyze it with the static method? How it was done?
Okay. Thank you for the very practical question. So we did the protein concentration measurement with BCA kit, and we also measure OPA. So we know like free amino groups, and we also know the protein concentration. So we did both way.
Okay. Thank you very much. I think we have one more question. Do you want to go for this? For this one?
I'm going. Yeah, it's more a personal opinion question here. According to your experience, what's the pros and cons you find with working with MRI, and how much can it be reliable, you know, to validate this in vitro, in silico models? How far away are we into it? You know, what's next? What's in the future?
Thank you. I think this is also a very interesting question. And if you ask me about how I think of MRI, I find it amazing, first of all, because I think it is super cool that it's an approach that we can do both in vitro and in vivo. So we can apply the same way. In that sense, it already compared to many other approaches, it already give us the convenience that we can directly compare these two measurements in vitro and in vivo, because if we can apply the same way. So that's one of the advantage why I think it can be useful to help us to validate in vitro models. Plus, because with MRI, it's a non-invasive model, non-invasive approach. So we can try to measure many things, and with understanding what's happening in the stomach, for example, then we can better understand the dynamics of the stomach. Then we can maybe use this information to link with how different food property affect the gastric dynamics, and using this as input to help us to validate this in silico model or in vitro model. So I think that can be one of a very good application in the future. And what else you asked me? The disadvantage and advantage of using MRI.
The pros and cons, yeah.
I can only talk from my personal point of view. In the beginning, I find it quite difficult to work with MRI because I had my education background as a food scientist before my PhD. I didn't do higher, like more intense physics before. So it took me a while to try to understand the concept, because if I don't understand anything, it's really hard to interpret the markers. So that's one of the I wouldn't say disadvantage, but I would say one of the challenge in the beginning when I faced with when working with MRI. And the nice thing of working with MRI is that then you would start to learn the new things. And what I find is people who work with MRI are all very nice and very helpful when I discuss with people. So that's one of the things that I like about. And another thing is that because beforehand I was more working in the lab with in vitro samples. So when I did my human trial, I also find it very interesting to interact with people, and learning how to operate the machine is it's also cool. It feels like I'm a doctor or something. So that's in general my personal point of view. But in summary, I think it's a very good approach, and there are many possibilities, and it surely deserves our attention and more effort.
Thank you very much for this sharing of experience. You know, it's PhD is a long time, you know, long experience. You master it now, and it looks amazing, honestly, when you present it.
Thanks.
And I think we are running out of questions for the moment from my side. Let me double check from the attendees. Talita, good. So I think we will thank you a lot, Ruoxuan, to have prepared this presentation, share your experience with us. And Talita will share a few slides to present the next webinars and few informations. I wish you the best for the future and your postdoc in Muireann at the moment.
Thank you so much.
So we'll have a next webinar next month, so the 1st of March, and we will welcome Elise Chartron from INRAE France. We'll discuss about human milk versus infant formula, protein digestibility and impact on microbiota gut-brain axis. Then in April, Marcos Infantes Garcia will from Leuven, Belgium, from gastric to small intestinal lipid digestion, the role of all-in-water emulsion design to affect in vitro lipolysis kinetics. And we are still waiting for application for the webinar of May the 3rd. So please feel free, you know, to spread the information and to email us at infogest.viva@gmail.com to apply if you just submitted, will submit your PhD over the last year. So a few information. So all the videos of the webinar series are available on the Infogest YouTube channel. And we have at the moment around more than 1,000 subscribers to the channel. You'll find all the videos of series with more than 300 views on each video, so which is pretty good for visibility of the presenters. You'll be able to find also information about the previous webinar and the Infogest protocol. Please don't hesitate to follow the LinkedIn Infogest channel. We have here around 1,500 subscribers, and we can thank the Infogest team for the follow-up. I wish you a Talita, so we are lucky to be together here. A very nice day, and we see you next month for the next webinar.
Thank you.
Bye-bye.
Bye-bye.